So last thing we need to take a look at is membrane transport. So this is just how we're going to get things in and out of the cell. So the plasma membrane is selectively permeable, which just means that some things can pass through the membrane and other things cannot. And they're going to need the help of a transport protein in the membrane. So there's two different types of membrane transport. We have passive transport and active transport. So I would definitely know this for the exam. So the difference between passive transport and active transport. So passive transport just means that energy is not being used and molecules are going to move down their concentration gradient. So what they're going to do is they're going to move from high concentration to low concentration. So I kind of like to remember this as you're going downhill. So you're moving from high concentration of the molecules to low concentration of the molecules. So they're going to move down their gradient, and that's why you don't need any energy because you're going down the gradient. And this will include simple diffusion, facilitated diffusion, and osmosis, which is just diffusion of water. So anytime you see the word diffusion, you know that it's going to be passive transport. And then active transport is pretty much the opposite. So you are going to use energy, and the molecules are going to move up their concentration gradient. So they're going to move from low concentration to high concentration. So they're going to go up their gradient. So they're going to go up the hill. And this will include active transport, endocytosis, and exocytosis. So let's start by looking at simple diffusion first. So simple diffusion can be done by small hydrophobic molecules such as oxygen and carbon dioxide. And what these molecules are going to do is they're just going to move directly through the plasma membrane, and they're just going to move from high concentration to low concentration. So they just go right through the plasma membrane. They don't need any help of any transport proteins. So that would be important to know for the exam, just how simple diffusion works, and that the molecules are just going down their gradient, no energy is used, and so I would know that. Okay, facilitated diffusion. Facilitate just means to help. So we've got some types of molecules that cannot cross the plasma membrane by themselves. So these would be hydrophilic molecules, for example, like glucose or sodium ions, and they're going to need the help of some plasma membrane transport proteins in order to get across. And so this type of transport, we have got diffusion in the name, so we're not going to use any energy, and the molecules are going to go down their concentration gradient. So I've got a couple pictures here. So if we look over here on the right, we've got sodium ions going from high concentration, going down through this transport protein to low concentration. So moving from high to low concentration, they're just going down their gradient. No energy is needed to do that. And then here on the bottom left, we've got a little animation of facilitated diffusion. And you can see we've got high concentration of, maybe this is glucose inside the cell. And then we've got lower concentration outside the cell. And then we've got this transport protein in the molecule that is just flipping the glucose from high to low concentration. So again, we're going down the gradient and no energy is being used. So that's facilitated diffusion. Okay, osmosis is just the diffusion of water through a semi-permeable membrane, for example, the plasma membrane. And water can cross the membrane, but the solutes, the things that are dissolved in the water, maybe it's salt or glucose like sugar or something like that, they cannot cross the membrane. And water is just going to move down its concentration gradient from high to low water concentration. And no energy is going to be used in osmosis because it's just diffusion of water. Okay, so here we've got a picture of water, and we've got the solute molecules here in purple. Maybe this is salt or something. And we've got a semi-permeable or selectively permeable membrane in between the two halves of the tube here. And you can see we've got less solute on the one side and more solute, more salt on the other side. And when you have less solute, that means you have more room for water. So we'd have a higher water concentration on the left side. And then on the right side, we've got more solute, which means we have less room for water, so we have a lower water concentration on the right side. So water is just going to move down its concentration gradient and going to move from high to low water concentration. So the water will just move from the left over to the right side. And so you can actually see that the water level actually drops as the water moves over to the other side. Okay, and then we have active transport. So remember, active transport is very different. It's the opposite of passive transport. So we're going to be transporting our molecules up their concentration gradient, going from low to high concentration, and we are going to be using energy. And for this one, we're going to be transporting hydrophilic molecules, maybe like glucose or sodium ions or something like that, that can't normally cross the membrane, but we're going to use the help of a transport protein here. And you can see that we're going to go from low concentration up the gradient to high concentration, and you can see we're going to use some energy here to do this. So we're going to use some ATP to do that. So that's active transport. Okay, and then our last two, we're going to use a vesicle, a little membrane-bound sac, to move things across the membrane. So with endocytosis, endo means inside, so we're going to be taking things into the cell. Okay, so we're going to have our cell reach out and surround with its plasma membrane and bring in that particle. And then here it's just showing fusing with a lysosome filled with digestive enzymes and then digest what was taken inside. So endocytosis, we're going to be bringing things into the cell by reaching out and surrounding it with a plasma membrane, and then just kind of pinching it off part of the plasma membrane and then bringing that into the cell. So that's endocytosis. And then exocytosis is just the opposite. So exo means outside, or I just remember X for exit. And this is where we're going to take a little vesicle, a little membrane-bound sac filled with something inside the cell, and it'll come up and fuse with the plasma membrane and then release its contents to the outside of the cell. Okay, so this is exocytosis. So we will use this to release like neurotransmitters from our nerves. This is how the milk in the mammary glands actually gets dumped into the ducts of the mammary glands. They get released this way. This is how antibodies made by our white blood cells get released from the white blood cells. So there's a lot of things that are released this way. Hormones, like protein hormones like insulin, get released from the pancreas this way as well. So there's a lot of ways that exocytosis are used by the body.